Welcome everyone to the 2024 Biophotonics Conference. Thank you all so much for joining us, and in this session you will hear from Slava Artyushenko, the founder and a senior advisor of Art Photonics, and Nazanin Shafiee, the director of strategy and business development at Avantes. Together, they're joining us to introduce viewers to advanced fiber optics for live spectroscopy applications in medicine. If you have any questions or comments, you can type them right into the conference chat box that is on your screen. And if you missed part of today's session, you can always come back later and watch it on demand. That's all from me, Slava, Nazanin. We appreciate you both for taking the time to join us at the 2024 Biophotonics Conference.
I am proud to be invited second time to be a speaker for Biophotonics Conference. And I would like to go through my slides to show how our history of 25 years, today it's realistically 26 years, was really developed for the application in industry and medicine. I am starting from my relatively old slide, but it's really important, at least for the picture over here, where you see the trend in medical world, that it's more and more diagnostics from 2007 up to 2025. Because before you start any kind of intervention, you should know exactly where is it and how to solve it. And very important figure also this nice round diagram that biophotonics in general is one of the major, or simply the major, let's say, segment of human activity business, because all of us, we would like to be healthy and young for 100 years old or 150 years old, and that's why our work was devoted to this kind of nice purpose. Our specific advantage, I should say, for company Art Photonics, which I have started in '98, is that we try to use any type of fibers, silica, infrared glass, polycrystalline fibers, even hollow waveguides, to cover very broad spectra. Our motto is broad spectra fiber solutions from UV up to mid-infrared range. And when we were looking for medical application, we try also to develop very tiny probes. And our smallest diameter probe is Raman probe, developed together with a partner in Holland, company Reverdi, which is smaller than 160 micron in outer diameter. My baby is this mid-infrared fibers, which cover very broad range, so-called fingerprint region. Advantage of transmission for polycrystalline fibers, we produce in-house from silver halide crystals, that they cover the range, which is in excellent match with infrared emission of human body at 330 degrees Kelvin. So in result, it's possible, it's opened the way to do so-called infrared endoscopy. And we were proud to work with Boston Scientific, with Securus for such type of project, where you see our probe going through the esophagus to see operation on heart, which was realized with radiofrequency ablation catheter. It was very good patent from Securus and then from Boston Scientific. So we were able to see hot spot during the procedure inside a human heart. But to be honest, we really devoted most of our applications to spectroscopy, because in general, you can cover, of course, laser power delivery with fibers, you can make infrared pyrometry, but mid-infrared, Raman, and infrared spectroscopy will give you a chance to see real composition of tissue or bioliquids. In mid-infrared, it's excellent spectra. It's well-known absorption bands, and it's much better expressed compared to the first harmonic, first overtone of the same vibrations of molecules. And that's why over here, silica fibers are transparent in this blue zone. but unfortunately they are non-transparent after 2.2 microns for longer wavelengths. But we have all. We have mid-infrared fibers, we have silica fibers, and Raman spectroscopy is also about vibrations, but it can be realized with silica fibers. Silica fibers are used for many applications already. It's chemical, petrochemical, pharma, and historically, our very first request was really how to control fermentation during brewery. That's why we developed very special probes for that. It's named transflection, so light goes to the mirror and then return back. But for food industry and biopharma, there are very strict demands on polishing, on welding, soldering of the optics, Sapphire optics over here. And we are proud this year, it's our news for this year, that we got so-called 3A certified certification for our probes, together with our partners, Spec5 USA. They help us to develop them. That's why now we can use it with food industry, including brewery. Mid-infrared range, I would like to show, it's my, how to say, pleasure to introduce my very good partner and friend, Professor Abraham Katser, who tried to detect melanoma and other diseases on the skin using mid-infrared fibers. And you see the range is between, let's say, roughly speaking, 9 microns to 10 microns. And this is very clear difference for the melanoma, which is red, and normal tissue. So that's why here I really can say that there is less problems now to detect melanoma because classic way is just to look for it, to make a biopsy and wait for 1 or 2 days. Patient is nervous, while spectroscopy way can give you a chance to get an answer within few seconds, minutes maximum. Our approach for cancer diagnostics, especially to see where is the border of cancer, so-called tumor margin, was really to look for all methods of spectroscopy, all key methods, fluorescence, near-infrared reflection, mid-infrared absorption, Raman. Why? Because in general, most of the scientists and companies, they look only for one method, while complementary information from different methods can provide much more precise information. For example, you can see when it's really only one method, then you will see so-called false negative and false positive reaction. When you combine data from 2 methods, it's much better separation, and surgeon can really distinguish where is the border between normal tissue and tumor tissue, malignant tissue, to make less invasive but fully radical removal of the tumor. Again, one slide to show that all fibers we use covers this broad range from, let's say, 250 nanometers up to 17 microns. And they are compatible with different spectrometers. This is FTIR spectrometer from Mettler Toledo, Raman spectrometer from Vasage, 2 spectrometers from Ocean Optics, similar one from Avantes. We can also provide special couplers to use laboratory spectrometers with fiber optics for real-time live spectroscopy. Again, the same picture, you know, to show we started historically from renal cell carcinoma. It's a tumor of kidney. And you can see that one method will give you some kind of, how to say, confusing data. It's not clear where is the margin, but as soon data from 2 methods are fused, then it's much better separation of normal tissue and tumor tissue. We even developed some kind of provisorish, let's say, draft software to help doctor to see or different color of the lamp, or even to hear, let's say, different sound coming from the system. Of course, combination of mid-infrared spectroscopy and fluorescence, it's only one case. It also helps when it's combination of near-infrared and fluorescence, or near-infrared and mid-infrared, so to provide more precise separation of different tissues. There is one example which is for food industry. It's not for cancer, it's just a task for the chicken plant, where robots must really distinguish liver, breast, and fat. Mid-infrared spectra will give you some data, but still a couple of organs are overlapped. Fluorescence will also help, but nevertheless, again, overlapping. Only fusion of data from two methods will give you very clear separation of all three organs. Even more, it can be selected one wavelength in mid-infrared and one invisible to help for fast and low-cost separation of different organs, which is important for, let's say, conveyor, which is normally used at chicken plant. Our latest combination was realized for attenuated total reflection, absorption, and Raman spectroscopy. We united two silica fibers and two infrared fibers in the same probe. And in result, we were able to get Raman spectra or infrared spectra from different liquids, in this case, ethanol, acetone. But of course, with our interest to bioliquids, it's, for example, refined rapeseed oil. You can see both types of spectra to get, let's say, more precise composition of these liquids, of these mixtures. The most interesting was the highest combination we reached for one big pharma company in Europe, when they really asked to combine in one probe attenuated total reflection, absorption, near-infrared, and Raman. So, and this is very useful not only for pharma, it's useful even for milk industry, which is huge in the world. So you see how different is, for example, spectra for near-infrared for milk, fluorescence, and this is ATR-absorption. Raman is not shown here, but we did it as well. So that's why any process of milk treatment can be recorded in situ, in real time, in line, when it's done with control by fiber optic probes and proper spectrometers. With a focus on oncology, to help doctors to define, to distinguish normal tissue and malignant, we started excellent project. It was even awarded by Eurostar program, like one of the best, how to use Raman for it, Raman spectroscopy. And we did it with our very good partners, company River-D in Rotterdam, with Erasmus Medical Center in Rotterdam. Now we work close to the University of Antwerp. And idea was very simple. There is very interesting biomarker we found for oral cancer, squamous cell carcinoma, that for the normal tissue, ratio of lipids, a ratio of water is like that. Look for this green picture. As soon, unfortunately, tumor starts to grow, then concentration of water starts to grow as well. So measuring the ratio of these two peaks, we can distinguish border of tumor with a precision of 1 millimeter. When this tiny, the smallest Raman probe goes into the tissue with a speed of, let's say, 1 centimeter per second, we can really distinguish every 100 milliseconds where is the tumor and where is the normal tissue. But in general, I should say that all this kind of research work, of course, took time. We spent not 1 year to develop several methods, but when it's clear application, and for example, for let's say, medical purposes, you would like only to see water, then it can be done for medical purposes, for prostate cancer, we tried. It works even for powders, for starch, for sugar, because it's enough to have only 4 LED in this system and 1 photodiode to measure concentration of moisture with really high precision, like 0.2%. So it's not only one solution, it can be, and this is much cheaper. It's about, let's say, 10 times cheaper compared to any kind of spectrometer for the full range. There are also other solutions, very interesting one from Multispectra with 16 detectors and broad light source, which can be also fiber optic coupled. And our latest work published this year was really based also on idea not to use a full spectra, but to use only several wavelengths, which we selected for osteoarthritis diagnostics. You can see here, it was our first prototype, 7 quantum cascade lasers coupled into the fiber, and this fiber probe can go into the knee and measure which cartilage is healthy and which part is really already with osteoarthritis. So concept of this shown here. So you can really unite different wavelengths because quantum cascade lasers gives you very narrow, very good, very strong signal at distinct wavelengths. But we can combine them in any set selected specially for disease. For example, for osteoarthritis, we selected several reference bands and several signal bands. And in result, it was possible with 7 QCL in our second prototype. All QCL were really covered by this box, and you see only, let's say, one fiber which collect 7 wavelengths and go into the knee, so to the end of this probe. And then, it's only one detector which measure, the signal coming from each of these wavelengths. So that great project was named Miracle, made with 14 partners, including Art Photonics. And we are really looking for the future of this kind of solutions, which will be not only for the liquids or tissue, it can be also used for the exhalation monitoring, because diagnostics can be done for, let's say, control, during the control of exhalation of the patient. So just to end up, I would like to say that our interesting way into the application of fiber optic spectroscopy for diagnostics is really moving fast, but I think we are only in stage to open the door to the great future, where it will be not only research spectrometers used for research, it will be dedicated fiber optic sensors for specific diseases. It will be cheaper and more of it. Each one will have IP address and can be used like Internet of Things, which will enable even telemedicine. So diagnostics can be done at the other side of the planet when a real good doctor can make his conclusion looking for results from Singapore or vice versa, from Boston to Europe. So that's why I'm proud to be in the team of great partners to develop in the future more and more interesting solution. And final news to share that we are now members of Nazanin group, altogether 13 partners. So Art Photonics was acquired end of last year. And next presentation will be done by our colleague from company Avantes. And they are 30 years in business, even more than 30, we are 26. So that's why Nazanin will make a next presentation.
Thank you, Slava. Now I'd like to shift gears from the advanced fibers you just talked about to spectroscopy and its application in biomedical and life sciences. I'd like to take a moment and talk about who we are. Avantes is part of a photonics group called Dynamics, a group of companies with a collective mission to make the world a better place by implementing innovative photonics technologies to measure vital parameters across many fields, including biomedical and life sciences. And within the entire value chain of non-contact optical measurement technology, we can cover everything from components and subsystems to other applicable solutions. Before I dive into the details, let's first put things in perspective and look at the global healthcare market. As you can see from the numbers on this schematic, significant growth is expected from $512 billion in 2022 to a projected $665 billion in 2028, which is just 4 years from now, reaching $800 billion only 2 years after that. Now, this growth is being driven by several factors, including advancement in technology over the past few decades, the unfortunate rise in chronic diseases, the aging population, but more so one of the key drivers is the increasing demand for personalized and precision medicine, which is pushing and will continue to push this growth. We can expect significant development across multiple sectors, including pharmaceuticals, medical devices, biotechnology. And I'll focus on the relevant ones in my upcoming slides. Now, let's zoom in on the global biophotonics market. What we observe is that a significant segment of this market is focused on medical diagnostics and screening. This represents a notable portion of biophotonics market, which aligns with the trends we see in the medical community in the related sectors. The emphasis is on early diagnosis and treatment, and the focus is intensifying the demand for technologies as well as their integration into the medical devices. With that, my goal today is to discuss the technical possibilities and capabilities of spectroscopy. I'll highlight some state-of-the-art applications as well as some generic applications that we've either already been exposed to, perhaps without realizing that spectroscopy played a role, or are likely to encounter in our lifetime. Most importantly, I hope to foster further collaboration, as the successes you'll see in my slides are the result of the efforts of many teams and diverse expertise coming together from around the globe. Now, when it comes to spectroscopy and diagnosis, what we're trying to distinguish is between healthy and diseased tissue. Regardless of the method we use, whether it's reflection, scattering, diffuse scattering, absorption, fluorescence, Raman, the spectrum produced can enable the user to differentiate between what is considered normal and what is diseased. One of the notable advantages of spectroscopy is that it is non-invasive and can enable this analysis in real time. With the applications I'll present, there's plenty to discuss, so I've selected a few mainly to cover the breadth of possible applications, and even with this selection, there's much I won't have time to cover today. So let's get started. Let's talk about the brain. The brain is one of the most essential organs in the human body, and although it accounts for only 2% of the total body weight, the cerebral blood flow volume represents 15 to 20% of the cardiac output, which is roughly 20% of the body's overall oxygen consumption. In a 6-year-old, this figure is about 50% of the total body oxygen consumption. So cerebral blood oxygen saturation, or cerebral blood flow, CBF, has become a critical metric in surgical anesthesia, neonatal monitoring, clinical treatments of patients undergoing traumatic brain surgery. Now, this requires advanced NR spectrometers as a core engine of an oximeter. Now, if you look at this schematic on the right, what you'll notice is that it's not just one number that's important. We're actually examining the oxygenation level of the brain tissue, the regional tissue, and the amount of dissolved oxygen present in the jugular vein. Now, while most of us may not have needed cerebral monitoring, chances are we've all visited a medical facility and benefited from pulse oximetry technology, which provides non-invasively and in real-time blood oxygen saturation measurement. For caregivers, it's crucial to be aware of any changes in oxygen saturation as early as possible and to know these changes as accurately as possible. Now, the accuracy of some of these devices can be impacted by several factors, such as poor circulation, cold extremities, skin pigmentation, nail polish, and even strong ambient light. It's worth noting spectroscopy is often used to validate and qualify these devices and their components, because full spectroscopy sampling is not typically necessary for these simpler devices. However, depending on the use case, such as during surgery, one may want to consider more accurate setup. Now, what you see in the second picture on the left is an oximeter, which is measuring blood from a carotid artery rather than from a peripheral blood vessel. So because of the shorter distance, we're able to get much faster response to the actual change. Skin is the largest organ of the human body and serves as a protective barrier against physical, chemical, and biological factors. Considering its functions and how it's constantly responding to various stimuli, be it intrinsic or extrinsic, it's been studied a lot. Here are a few literature sources that focus on extrinsic factors such as cosmetic, pharmaceuticals, and nutraceuticals. The intrinsic factors have also been studied, and that's typically been done by deep tissue reflection. This method can be employed to detect cancerous tissues as well as assess the effectiveness of radiation therapy in reducing cancerous masses. Now, staying on the topic of skin, let's discuss something called skin autofluorescence. There are two major causes of accumulated damage during aging. One is oxidation and the other is glycation. Glycation is a chemical process in which a sugar molecule binds to a protein or lipid through a series of reactions. This process leads to formation of what's known as advanced glycation end products, or AGEs, which accumulate in various tissues, including blood vessels, muscles, bones, joints, indicating harmful processes that are occurring in the body. These harmful processes are linked to diseases such as cardiovascular disease, fractures, osteoarthritis, and these diseases can be influenced by exogenous factors such as a high-fat, high-protein diet, smoking, but also endogenous pathologies such as diabetes and chronic kidney disease. Having any one of these conditions can expedite the accumulation of AGEs in the human tissue, and by monitoring skin autofluorescence, the treatment of patients can begin at an earlier stage. Another way we can monitor the skin using spectroscopy is through a technique called diffuse reflectance spectroscopy, or DRS. DRS is a non-invasive or minimally invasive technique, depending on how it's implemented, that can be applied to monitor tumors and their response to therapy. It's compatible with the biopsy port of standard colonoscopes, which makes it very attractive for providing functional information to guide clinical decision making. DRS has been integrated into fiber optic biopsy needles to assess functional tissue properties in vivo, and if combined with pulse radiolysis, it allows for determination of time-resolved spectral changes in radiation therapy. There's a lot of work that's been done at the Irvin Beckman Laser Institute using this technique, and it's worth noting that it's shown an accuracy of 95% in differentiating the tissue type. Organ perfusion or blood perfusion is a crucial medical diagnostic procedure for assessing both normal and pathological physiologies, and it's essential to determining the viability of tissue transplants. Now, dynamic organ preservation is a relatively old technique. However, machine perfusion technique has only been implemented in the last decade, with the first clinical series occurring in 2010 for liver transplant. Now, for major surgeries, maintaining perfusion is vital as it provides real-time monitoring of tissue health prior to organ transplantation. And again, with this type of application, we work with partners who customize it for different settings. Now, let's move on to something that we will likely experience in our lifetime: osteoarthritis. Osteoarthritis is the degeneration of joint cartilage or the underlying bone, affecting synovial joints, which is often associated with pain, cartilage erosion, immobility, and general joint dysfunction. Degenerative joint conditions are typically characterized by changes in the properties of articular cartilage and subchondral bone. Visual assessment during arthroscopic surgery doesn't provide information about the subchondral bone integrity. However, it's been shown that, again, NIR has been effective is simultaneously monitoring the progressive degeneration of both cartilage and subchondral bone in vivo. Let's move to another application, co-oximetry. Co-oximetry enables the quantitative measurement of blood parameters, including oxyhemoglobin, deoxyhemoglobin, carboxyhemoglobin, methemoglobin, as a percentage of total hemoglobin concentration in the blood sample. Now, let's put that aside. Leading cause of death from unintentional poisoning is carbon monoxide, and early symptoms of carbon monoxide poisoning include headache, nausea, confusion, shortness of breath, weakness, loss of muscle control, symptoms that are often mimicking flu, leading to either misdiagnosis or delayed diagnosis, which can result in delayed neurological damages or death. Traditionally, these blood gas parameters are measured using a spectrometer via transmission or absorption. There are many advantages to using co-oximetry, including the absence of sample preparation, ease of handling, portability, all of which make it very ideal for diagnosis in forensic application. Now, before I discuss the specifics of oral mucosa screening, I'd like to say a few words about the rise of cancer worldwide, particularly among young people. The notion that cancer is a disease of the elderly is no longer accurate. The incidence of many cancer in young people is increasing at an alarming rate globally, which has created a significant unmet need for treatments targeting this younger population. The number of diseases affecting oral mucosa alone has increased by more than 20% over the past decade. Now, there are many possible techniques for diagnosis, but they each come with their own disadvantages. For example, visualization can be subjective, and it's difficult to detect early-stage disease. Immunological methods, they require expensive tools, and the minimum concentration level for some of these biomarkers can be detected at the point that the disease has already progressed to advanced stages. Fluorescence, well, some argue that this method lacks sufficient sensitivity and specificity in some cases. Now, histological analysis via biopsy is the most reliable method for diagnosing cancer, but it's very invasive, and it places additional demand on the medical system and requires highly qualified doctors. Again, DRS is a technique that can be very attractive in early detection in tissues. It's also not invasive, and it provides answers in real time. One last slide on application is about pharmaceutical process monitoring. Now, FDA's Process Analytical Technology Initiative forms the basis of pharmaceutical good manufacturing practice, the GMP rules, for the 21st century. Now, pharmaceutical industry is a highly regulated industry with products that must meet very stringent specification. But this doesn't necessarily mean that pharmaceutical processes are all very optimized. Now, conventionally, pharmaceutical manufacturing used batch processing, which involved offline, time-consuming, and less efficient laboratory testing to evaluate the quality. Nowadays, optical spectroscopy, including NIR and Raman spectroscopy, is widely used across the industry for real-time processing measurement. That's actually for pharmaceutical, food, and many other chemical and biochemical industries. Now, these techniques can be applied at-line, on-line, and in-line to monitor process effectively. And with Raman specifically, we can have detection limits as low as ppm and very low prediction errors. Let's briefly talk about a few requirements for medical devices from a subsystem perspective. We live in a very noisy world. The ability of equipment to operate satisfactorily and without adding to this noise is crucial. This concept is captured in electromagnetic compatibility, which essentially encompasses the two major requirements. One, the emission of the instrument, ensuring it doesn't degrade the performance of nearby devices, and two, the instrument's immunity or tolerance to intended and unintended electromagnetic environment. Now, EMC standards in medical environment are a lot more stringent than in other fields because lives can be at stake. So in 2022, the FDA actually released new guidelines to ensure patient safety, and Avantes products are among the few on the market that are EMC compliant with these new requirements. I'd like to add a few words about our manufacturing facility. We utilize custom semi-automated processes to enhance modularity, reproducibility, and product performance, while also enabling the upscaling of production volume. These features of our future-proof manufacturing process result in high unit-to-unit reproducibility with predictable throughput, allowing Avantes to deliver a wide variety of spectrometer configurations reliably. Now, reproducibility that I refer to doesn't apply to products within a series, but also products from batch to batch. And we achieve this by integrating modularity into the concept, along with semi-automated systems, integrated quality control, and the use of statistical process control. Now, I have presented a selected number of spectroscopy methods and their applications in diagnostics and monitoring. Comparing these methods will enable the selection of the best option, whether it would be the fastest, most sensitive, specific, or accurate for detecting diseased tissue. For cancer, the choice will depend on the organ and type of cancer. Ultimately, what I hope you take away from this discussion is how these spectroscopy methods have become much more powerful tools, increasingly used over the past decade with significantly higher accuracy for real-time diagnosis and screening. As a global community, we're still far from using and leveraging spectroscopy in all possible state-of-the-art diagnostic and monitoring applications. I hope that through platforms like this, we can foster greater collaboration among different teams globally. And with that, I'd like to invite you to connect with me personally or with our team to continue the conversation. Thank you for your attention.
Thank you, Slava and Nazanin, for joining us to detail advanced fiber optics for live spectroscopy applications. To everyone joining us in the audience, I have one final reminder. If you haven't already, you can type your questions and comments into the conference chat box, and do not worry, if you've not been answered yet, you will be reached via email at a later date. This session is hosted by us at Photonics Media, and we hope you're enjoying the 4th annual Biophotonics Conference. Thank you all so much for joining us.